exchange. Nutrient and gas exchange in the capillaries. That's the next thing we want to talk about. So it's a diffusion concept. Are we comfortable with diffusion? Remember our rules of diffusion? Hypertonic, hypotonic, remember those terms? Okay. So diffusion is a gradient-based phenomenon, where if you got things at a high concentration, they tend to diffuse to a low concentration. Okay. So there's two things we want to keep an eye on here. And the diffusion gradients, we're not going to concentrate on too much right now. It's going to be really important when we talk about respiratory function. But suffice it to say, that is how cells exchange nutrients via a diffusion gradient. We also have pressure gradients, and that's a little different. We haven't really talked about that before. For respiratory gases, the way a diffusion gradient is expressed is not so much in concentration, but in pressure. Okay. We have a pressure of oxygen. It's higher in one spot and lower in another spot, and gases diffuse according to their pressure gradient. Okay. Well, in all cases, though, capillary beds are fed by arteries and drained by veins. Okay. In all cases, capillary beds are fed by arteries and drained by veins. So in order to get bulk exchange, bulk nutrient exchange, what we have to have happen is we have to have a little bit of the fluid of the blood leave on the artery side, bathe the tissues in that what we call interstitial fluid at that point, where we can have the nutrient exchange happen, and then we got to drain that fluid back into the veins. We call that bulk flow. So going into a capillary bed, we have to have some of the fluid, just a small percentage of the fluid, leave the blood, go into the interstitial space where we can have the diffusionary process of nutrient exchange occur. And then we got to drain that fluid back into the veins for return to the heart. Okay? And this is a pressure-driven process. We're going to have to talk about the pressures involved here. Here's a picture showing us a fenestrated capillary. So this is the kind of capillary that might be doing nutrient exchange. Probably not sinusoidal, because those are the really big, super leaky ones that you find in things like lymph nodes and the liver. But a simple fenestrated capillary, which is what you would find in pretty much any organ. What layers do we have to diffuse through? Okay, what layers do we have to diffuse through? So here we're in the lumen. There's those red blood cells. We have got a blood vessel with a diameter of about 10 microns. Well, we got to go through the apical surface of the endothelial cell, basal surface of the endothelial cell, then the basement membrane, which is what this is, and then finally we're out into the interstitial space. So we have to diffuse through those layers. You've got two choices. You can go the transcellular route or the paracellular route. Transcellular means moving through the cell. Paracellular means going around.
These capillary beds don't have like the tunic, intima, and tunic media.
No, capillaries do not have the three tunics. Capillaries only have endothelium. Okay. That's the hallmark of a capillary. It does not have any muscle around it. There's no tunica media. And the other terms I gave you, those are just terms that I want you to understand. If you go through the cell, it's called transcellular. If you go around the cell through the lateral surfaces, that's called paracellular. But we really aren't going to do anything with those terms until we get to renal function. We'll bring those up again.
What did you say they have? They don't have tunics, they have...
Only the intima, which is the endothelium. Capillaries only have an endothelial layer, which is equivalent to the intima. Okay, so the pressures that establish or control bulk flow. Here's a diagram for you. We've got four pressures. Two are hydrostatic pressures and two are what are called colloidal pressures. So there's a slightly different set of terms that I like better than what this book uses, and I'll put them both on the board. And you'll see it doesn't matter which ones you use whatever's more comfortable for you. What's a colloid? Anybody remember? Like Jello, I heard somebody say? Okay. Well, the definition of a colloid is a solution that has relatively large solute particles that never settle out, that can...
What kind of particles?
Large solute particles. And it can pass through a filter but not a membrane. Colloids can pass through a filter but not a membrane. And I thought I heard somebody say Jello. Yeah, Jello is an example of a colloid. Okay. The other two terms to have in your mind when you think about a colloid, sometimes it helps to know what it's not, right? A true solution, which we usually talk about aqueous solutions, solutions in water, like salt water. Okay. The difference there is that solutions can pass through a membrane. Colloids can't. Okay. And the third type of mixture that we deal with when we talk about this is a suspension. And a suspension has really large particles, but the thing is there, in a suspension, the particles will settle out over time. So something like muddy water is a good example. You let that sit for a while, and you'll come back and you'll see the fine sediment on the bottom, and then just clear water, right? So here's an important point to file away, and I never asked this on a test, but it's news you can use. Medications that you buy that come in liquid form are almost always suspensions. Almost always suspensions. And that's why they usually say, shake before using. Okay? If you don't do that, and it's sat in your fridge for a couple of weeks or something like that, all the active ingredient is settled out on the bottom, and all you're taking is some syrup, okay? You gotta mix those to get the active ingredient resuspended.
That's in a solution?
That's in a suspension. And the reason I threw those three terms out now to review is that blood is an example of all three. Okay? When we dissolve things like electrolytes and salts in the blood, well, that's a true solution. Okay? Sodium dissolved in blood, that's a true solution. That will never settle out and then go through a membrane. But proteins in blood, that's a colloid. Okay? That's colloidal. But blood is also a suspension, and that has to do with the formed elements of the blood, which are things like white blood cells, red blood cells, and platelets. With that respect, blood is a suspension. Those will settle out.
So you have colloid suspension, and what was the third term?
True solution.
True solution.
Yeah. All right, so your salts and electrolytes in the blood, that's a true solution. Your proteins, that's a colloid. And your formed elements of the blood, your blood cells, that's a suspension. So you got those three concepts going on. Right now, we're only interested in the middle one, the colloid concept. Okay. Now, when I have two solutions, and they have, now let's just talk about the dissolved proteins. They have different concentrations of solute. That sets up a gradient. Okay. And here I'm thinking about the dialysis lab that we do in Bio 1100. I know most of you probably took that class. We take this little flimsy bit of dialysis tubing, we put some stuff in it, we tie off the ends, and we put it in a beaker, and we watch what happens over time, okay? And we're studying the process. Who remembers what process we're studying in that lab? Begins with an O. Osmosis, yeah, we call that the osmosis lab. Right? And the idea here is osmosis is the diffusion of water through a membrane. Remember that definition? Diffusion of water through a membrane? Osmosis always diffuses into the hypertonic solution. Whatever solution is more concentrated will draw water to it. So in the cardiovascular system, we're interested in the osmotic pressure of the blood, which is primarily due to the colloidal content of the blood. So we call that blood colloid osmotic pressure, BCOP. Okay? Blood colloid pressure, BCOP. Now your book calls this osmotic pressure capillaries? Right. Same thing. I think it's 20. We use 26. Okay. We'll use your book's examples. All right, 26. The unit I'm using there is millimeters of mercury. That's a pressure unit. Okay. Like atmospheric pressure is 760 millimeters of mercury. Okay. All right. The interstitial fluid. So remember what's going on here. We've got a blood vessel, and the blood vessel is traveling through a tissue space. So here are your tissue cells. So this is your blood. And out here is interstitial fluid. Okay. Do cells usually leave the blood? Do your white and red blood cells typically leave the blood? No, usually they stay in the blood. How about proteins? Do proteins typically leave the blood? Some. Mostly no. Okay, most of your proteins stay in the blood. Now salts and electrolytes and fluid leave the blood, but usually most of the proteins and all of the formed elements stay in there. Okay, so based on that, what do you think has a higher osmotic pressure, the blood or the interstitial space, or the interstitial fluid? Which one do you think has a higher osmotic pressure? Blood, a lot higher. Okay, so we've got blood colloid osmotic pressure, and then we have the interstitial osmotic pressure, interstitial colloid osmotic pressure. Here we'll just put pressure capillaries here. I think that's what they use on there, right? Oh, they put C. And then we have ICOP, which is osmotic pressure interstitial fluid. That's what they put up there. And what are they? Usually that's like 1. Yeah, that's 1. Okay, so we'll put, I'll erase that, and I'll just put, what do you think? Put it right under, will that make sense? Okay. And here we'll put 1. Okay, well, there are 2 of our pressures we have to deal with. Those are the 2 osmotic pressures. Now, if that were the whole story, which way would fluid want to move? Into the blood, out of the blood? She said out of the blood. I would disagree with that. Okay. Hypertonic, the more concentrated solution. Blood's the more concentrated solution.
Oh, yeah.
Osmotic pressures work by, the higher the osmotic pressure, that's the tendency to draw fluid to it. Okay. So in this case, if this were the whole story, vessels would just draw the fluid in from the interstitial space. It would work like a sponge. Well, think about what we need to do. On the artery side, if the blood is flowing this way, over here we have an artery, right? And over here we'd have a, what should be over here then? A vein. Okay. Well, we've got to get fluid moving out over here. And then we have to collect it back up over here. Follow the idea? Now, that's not blood. You're not having blood go out into the interstitial space. You're just having plasma, you're having fluid. Okay? So we're getting fluid flow out on the artery side and in on the vein side of the capillary beds. With me there? So if this were the whole story, the vein side would work fine. We'd be drawing the blood back in, which is what the veins want to do. But the arteries would be screwed. Okay? Because the arteries got to do the opposite. The arteries got to get fluid to go out. So we got to have 2 other pressures involved to take care of that problem. And those are our hydrostatic pressures. Okay, so these are colloid pressures here. That's why I had that written up there. And then we have our hydrostatic pressures. And hydrostatic really is just blood pressure. Well, it's mean blood pressure. Our 2 hydrostatic pressures that we deal with are hydrostatic pressure, I'm going to say blood or capillaries. Let's say A, C, C. Okay. Hydrostatic pressure, C. That means in the capillaries. And hydrostatic pressure, interstitial fluid. And for the blood pressure in the vessels, in the capillaries, I'm going to split that into 2 subdivisions. Artery versus vein. Okay? So we've got a certain amount of blood volume that's in our arteries.